Okay, so today we're talking about chapter 18. We're doing amino acid oxidation and the production of urea. So in this case, we're talking about how do we break down amino acids and get rid of unwanted amino acids that we have in our cells. So most of these amino acids are going to be metabolized in the liver as ammonia. So when we break down amino acids, the key or the problem is the nitrogen in the amino acids. So nitrogen is a special thing within our cells that we have to be able to process in a way that it does not develop into a toxicity or a toxic substance in our cells. And so that's what this chapter is really all about, is how do we handle that nitrogen and excrete that nitrogen in such a way that it is safe for our body and for our cells. And so most of that nitrogen, as we see in this chapter, is going to be converted into ammonia and then dealt with in a way that we can excrete it safely from the cells. So where do these amino acids come from that we're breaking down? So they can come from normal protein breakdown. So obviously we know that we can sometimes make proteins that we then don't need, that after they've done their job, we degrade them. And so so we can get normal protein breakdown and have those amino acids that we have to deal with. We can take in excess amino acids in our diet that we don't need. That if it's excess, then we have to get rid of them and break them down. Or if you are under conditions of starvation or diabetes, so that's when carbs become unavailable. That's when you start breaking down proteins and using those as fuel. So starvation, under conditions of severe starvation, or sort of severe diabetic conditions, when we start breaking down proteins as a source for fuel. That's where we get these amino acids. And then this chapter is all about how we process specifically the nitrogen in those amino acids and excrete it out of our cells. And predominantly, the things that we are talking about in this chapter are taking place in the liver. So we'll talk about how we do that as we go through this chapter. So here are our key topics for this chapter. So we'll talk about how proteins are digested in animals, how the amino acids are degraded, and then how urea is made and excreted. So this is the urea cycle that we'll talk about towards the end of this chapter. The first bit of this chapter will be more about how we process those amino acids, how we break down those proteins, and how we process those amino acids. All right, so we do use amino acids as fuel, but the use of amino acids as fuel varies greatly by organisms. So in carnivores, so these are organisms that obviously eat meat, about 90% of their energy needs are met by amino acids immediately after the meal. So they're taking in large amounts of proteins, and so they can meet the needs of their cell, the energetic needs of their cell, through the use of those amino acids and the proteins that they bring in. Microorganisms have to scavenge for amino acids from their environment for fuel when they need it. So they're not taking in massive amounts of protein that serve as that amino acid source. For herbivores, only a small fraction of their energy needs are going to be met by amino acids. So the plants that they eat are not a great source of amino acids. So really their use of amino acids as a fuel source is pretty minimal. Plants in general do not use amino acids as a fuel source, but they can degrade amino acids to form other types of metabolites, and we'll talk about that as we go through this chapter. So some organisms rely very heavily on amino acids as a source for fuel, other organisms much less so. All right, so what we have in terms of amino acid oxidation is if we have those leftover amino acids, so this is from normal protein turnover, normal protein breakdown. Once we have used a protein to do its job, we have those leftover amino acids. We have the dietary amino acids that if we've taken them in excess through our diet, more than we need to carry out protein synthesis, then we have to break down those amino acids. You can also have, again, during cases of starvation or severe diabetes, you have those proteins breaking down as a fuel source where we're using the amino acids for energy. So this is the source of the amino acids that we are breaking down, that we're oxidizing, that we're talking about in this chapter. All right, so dietary proteins are enzymatically broken down into their amino acids. So this occurs because of the acidic environment in the stomach. So we know that the stomach is a very acidic environment, contains enzymes that are responsible for breaking down proteins. So pepsin is a enzyme in the stomach that cuts proteins into peptides. It cuts every time there's an aromatic amino acid, pepsin makes a cut. So remember, this is your phenylalanine, your tryptophan, and your tyrosine. So every time there is one of those amino acids in your peptide, pepsin makes a cut and cuts that protein down into smaller chunks. Trypsin and chymotrypsin also cut proteins into smaller peptides based on that are located. So the trypsin and chymotrypsin are located in the small intestines, and so this comes from pancreatic secretions that come into the small intestines that work to degrade those proteins down into their amino acids. You also have aminopeptidases and carboxypeptidases that degrade proteins into amino acids further in the small intestines. So both these secretions, trypsin and chymotrypsin, and aminopeptidases and carboxypeptidases, come from secretions that come from the pancreas that lead into the small intestines that work to break these proteins down. Now, obviously, when we are making cuts in these proteins to cut them down into peptides, we're denaturing those proteins, which leaves them open to even more enzymatic degradation as they travel through the small intestines and are further processed. Okay, so here's just a quick reminder for you of the digestive tract. These are things that you have talked about certainly in other classes, and just a good example of where these secretions come from. So here we have the stomach. So we know, again, that the stomach has a low pH. It's a very acidic environment. So this is where you get pepsin that begins to start digesting those proteins. When you eat, there is an enzyme called gastrin that is released, and that stimulates the production of pepsin, which is going to start breaking down those proteins in the stomach. And the lower the pH, the more activated that pepsin is. So you don't get pepsin that is activated in other areas of the body, but only in the stomach due to the low pH. You also then have your pancreas, which is here, sort of this yellowy structure here. This is your pancreas, which is going to release secretions into the small intestines, which is here. And so the pancreas is what's going to release the trypsin and chymotrypsin and the peptidases into the small intestines, which again further break down the proteins into their amino acids. All right, so if we take a quick look overall at amino acid catabolism, this is kind of showing you the intersection between some of the cycles that we have already talked about and the cycles that we are going to talk about. So here we have our intracellular proteins. So this is where we're breaking down those proteins that have done their job, and we're going to break them down into amino acids. We can also take in amino acids through dietary proteins. So those can also be broken down into amino acids. What we know about amino acids, remember, every amino acid has that amino group that has that nitrogen on it. And this is what we're really interested in thinking about amino acid oxidation and how we process these things. So the amino group is going to be key, and the first thing that we're going to have to do is break apart that amino group from the carbon skeleton. The carbon skeleton we can keep, we can reuse, we can recycle inside the cell. It is that amino group that has to go through nitrogen processing that we have to very carefully treat and get rid of so that it does not become toxic. So the carbon skeletons become alpha keto acids and can enter directly into or become components of the citric acid cycle and be used that way. So they get recycled, they take a different sort of pathway, different sort of course from that amino group. The amino group can be, a couple of things can happen to the amino group. So we can use it in the biosynthetic pathway of other amino acids, of nucleotides, or of other biological amines, other biological molecules that have those amino groups. So these are going to be particularly one of the things that we've already talked about are your pyrimidine synthesis, right? So we know that we need those amino acids. We use glutamine and aspartame in biosynthesis of the pyrimidine pathway. And so we need those nitrogen sources to carry those things out. The other thing, and so that comes in the form of carbamyl phosphate. So if you remember, this is an important precursor in the pyrimidine pathway. So that's one of the things that can happen to that amino group from the amino acid. The other thing that can happen also, so carbamyl phosphate sort of has two fates. It can be used in the pyrimidine synthesis. It can also lead into the urea cycle. And so this is what we'll talk about later in this chapter. This is where you end up producing urea, and that is your nitrogen excretion product. So if we don't need that nitrogen, it's going to be excreted in the cell as urea. Now you can see that these two cycles, the citric acid cycle and the urea cycle, are linked via this aspartate arginosuccinate shunt. Now we're not going to spend a lot of time talking about this pathway in particular, but I think it's interesting to see that these two seemingly unrelated cycles of the citric acid cycle and the urea cycle that you wouldn't think would have a connection actually do. And through this shunt pathway of the aspartate arginosuccinate, that we can shuttle intermediates from the citric acid cycle to the urea cycle and vice versa. Okay, so this is just a diagram sort of refreshing your memory of the citric acid cycle and how different amino acids can feed into that citric acid cycle. So again, this is not something that I would ask you to memorize, but this is just an example of showing you where all of these amino acids feed into that cycle and how we can link then the amino acid oxidation or the breakdown of amino acids to the citric acid cycle. So if you see, for example, things like arginine, glutamine, histidine, proline can be converted into glutamate, which feeds right into the alpha-ketoglutarate intermediate of the citric acid cycle. Things like isoleucine, methionine, threonine, valine, those can feed right into the succinyl-CoA intermediate of the citric acid cycle. So you can see that along this pathway, we can take care of just about every single amino acid in your cell and feed it into the citric acid cycle as a way to oxidize it and to break it down. Okay, so the first step in degradation of amino acids for all amino acid, for any amino acid, is to remove that amino group. So here we have our sort of generic structure of an amino acid where we have that alpha carboxyl group and the alpha amino group, the R group and the hydrogen. The first thing that we have to do, so remember back to a couple slides ago, we have to separate the amino group from the carbon skeleton. So the carbon skeleton is actually going to be used, remember, as the alpha keto acid, and that's going to go off and can be used in the citric acid cycle. The amino group is what we have to worry about or figure out what to do with. So the enzyme that catalyzes this reaction are either aminotransferases or transaminases. And so we'll talk a little bit more about these specific enzymes in a minute, but you've heard these words before. You've heard the names of these enzymes before as we've gone through some of the metabolic pathways that we've talked about. So it's we're either transferring we're basically transferring that amino group off of that amino acid to glutamate. So the ultimate goal of the degradation of amino acids is to transfer that amino group to a glutamate, to an L-glutamate molecule. We know that L-glutamate is used as an amino group donor in biosynthetic pathways or excretion pathways. So remember back all the way to chapter 22 when we talked about pyrimidine synthesis and purine biosynthesis. We used glutamate as that nitrogen source, as the donor, to build those nitrogenous bases. So the ultimate goal here is to get that amino group transferred to that glutamate group. Now, a couple of different pathways or options that can happen to that amino group within the cell is that amino group can ultimately be transferred to ammonia, urea, or uric acid. So most vertebrates are going to excrete their nitrogen as urea. Birds and reptiles are going to excrete their nitrogen as uric acid. And some microbes, some fish, they can excrete their nitrogen directly as ammonia into the water. So you can have high ammonia content in the water. So depending on what the organism is, that determines how we excrete that nitrogen. Now, this pathway, talking about glutamine from muscle and other tissues and converting that to alanine from other tissues, we're going to talk about that in a little bit. So that's called the glucose-alanine cycle, and we'll talk about that at the end of this chapter. So we'll just kind of hold on to that thought for a little bit, and we'll talk about that in a bit. Okay, so again, this is just kind of going back to that idea of the different organisms have different fates of nitrogen. So plants actually almost conserve all of their nitrogen. They don't really have a nitrogen excretion pathway or process. They actually conserve that nitrogen and use it in their metabolic pathways. Aquatic vertebrates, again, they can release their nitrogen directly as ammonia into the environment. It can pass through by diffusion through their epithelial cells and active transport through their gills. Terrestrial vertebrates, and as well as sharks, excrete nitrogen in the form of urea. So that's you and I. You and I would excrete our nitrogen in the form of the urea. It's far less toxic than ammonia. Ammonia is actually extremely toxic to animals. Urea has a very high solubility, so it will dissolve in water and is easily excreted through the kidneys in humans. Some animals, again, such as birds and reptiles, excrete their nitrogen as uric acid. As we know back from some of the other pathways that we've talked about, the degradation pathways of nucleotides, uric acid is rather insoluble. So it's going to form those crystals. It's not going to dissolve in water very well. It's excreted almost as a paste so that the animal actually conserves the water. Because we don't want to be excreting water as a general rule. So uric acid is excreted almost as a paste material to allow animals to conserve that water. Humans and other great apes, again, we mostly excrete urea, but as we know from purine breakdown, we can also excrete uric acid, and sometimes that can lead to problems like gout that we talked about back in earlier chapters. Okay, so here are those three excretory forms of nitrogen. So again, we have ammonia that we see in vertebrates and aquatic animals. We have urea that we see in terrestrial animals, including humans. And then we have uric acid, which we see excreted in birds and reptiles. So the key is that even though urea and uric acid contain carbons, you can see that these carbons are pretty well oxidized. They're very highly oxidized, almost entirely oxidized, and especially in the case of urea. Again, the idea is we only want to discard carbon after we have extracted all the available energy through oxidation. We don't want to be extracting carbon skeletons that still have hydrogens attached to them that we could still extract energy from. And so both urea and uric acid are fairly well oxidized to the point that we're not just throwing energy away. We're not excreting a whole lot of available energy from these forms. And so that's kind of the point of this whole chapter, is how do we extract all of that energy from those nitrogen sources before we excrete them. Okay, so again, the first step in breaking down these amino acids, especially once they reach the liver, is to remove that amino group by these aminases or aminotransferases. So here we are carrying out a transamination reaction, so we are removing the amino group from the amino acids. It's catalyzed by aminotransferases. We're going to use a pyridoxal phosphate cofactor, so a PLP cofactor, which you'll see in just a second. Typically, we use alpha-ketoglutarate as the acceptor of those amino groups. What that does is that leaves behind the carbon skeleton. So once we take off the amino group, we are left with those alpha-keto acid carbon skeletons, which again can sort of feed into the citric acid cycle or be reused and recycled in the cell. The point here is to collect those amino groups from lots of different amino acids, from all the different amino acids that we have in the cell, and ultimately get that those amino acids in the form of glutamate so that we can use them in other processes. L-glutamine is actually going to be sort of a temporary storage of that nitrogen, and that can actually donate the amino group when it's needed for amino acid synthesis. So we know we can also use glutamate in the form of the nucleotide synthesis. So glutamine and glutamate are going to be important here as we go through this process, and in sort of holding that nitrogen for us until we need it to be used in other processes, and then donating it when that time is needed. Okay, so this is the reaction showing that transamination of the amino group. So removing that amino group from an L amino acid. So here we have sort of, again, the generic structure of amino acid. We're going to pull that amino group off via the transamination reaction catalyzed by the aminotransferase. That pyridoxal phosphate as the cofactor, that's the PLP here. So we're going to remove that amino group. Alpha-ketoglutarate is going to be the acceptor. So when it accepts the amino group, it's going to form L-glutamate. So alpha-ketoglutarate with the addition of the amino group becomes L-glutamate. That is the goal here, again, is to get those amino groups in the form of glutamate so that we can then use them as nitrogen donors in other processes. What's left behind from the amino acid once we pull off the amino group is that alpha-keto acid. So that's the carbon skeleton that remains that can then go on and be used in the citric acid cycle. This is an easily reversible reaction too, by the way, so we can take that amino group off of glutamate and put it back on the alpha-keto acids to form amino acids when we need to. Okay, so what happens next is we have to take that glutamate that we just made, and we have to be able to extract that ammonia and process it for excretion. So this is happening in the liver. So this next step happens in the liver. We have to take that glutamate that we just formed in the cytosol, and we have to move it to the mitochondria so that we can process it. So the enzyme that is responsible for this processing is called glutamate dehydrogenase. So this is a huge enzyme. It has 6 subunits. There are lots of allosteric regulators for this enzyme. ADP is an activator of this enzyme, while GTP is an inhibitor of this enzyme. But there are lots of other regulation processes for this enzyme, which we'll talk more about at the end of this chapter. But again, the purpose is this glutamate dehydrogenase enzyme is only in the mitochondria. So we have to take this glutamate that we just made in the cytosol, and we have to transfer it to the mitochondria so that we can process it. So this oxidative deamination is what's going to happen here. And again, it's going to occur in the mitochondrial matrix because that's where the enzyme is. This enzyme, glutamate dehydrogenase, can use either NAD plus or NADP plus as the electron acceptor. This is the only enzyme that we know of that can use either one of these cofactors. Usually enzymes use one or the other, but not both. So glutamate dehydrogenase can use either NAD plus or NADP plus as the electron acceptor. So the ammonia that is removed, so again, this is a deamination, an oxidative deamination process. So we're going to take that amino group from glutamate and we're going to remove it in the form of ammonia. What we're left with then, when we take that ammonia group off of glutamate, is we regenerate alpha ketoglutarate. So this then can be reused and recycled in the cell. The ammonia is going to be processed for extraction in the liver. So we'll talk more about that in the next slides as we go through. But we're going to, basically what we're doing here in this process is we are collecting all of those amide groups from glutamate. We're collecting them as ammonia, and then we are going to be able to process them in the liver. Right now it's in the form of free ammonia, which is bad. Okay, we can't have free ammonia in our bloodstream, and so we have to do something with that ammonia. We have to process it, and in humans and other terrestrial animals, we process that ammonia further into urea for excretion. So the total pathway that we've talked about here, this transdeamination, we had the transamination, that was the conversion, the moving of the amino groups from the amino acids to glutamate. That was the first step. And now we've had the oxidative deamination. That's the second step. That's where we take that those amino groups from glutamate and we convert them into free ammonia, regenerating alpha ketoglutarate. So it's been a two-step process to get us here. So the transamination and the oxidative deamination. Okay, the key to this slide is really focusing on what we do with that ammonia that we just pulled off in the previous slide. So we had that glutamate, we pulled off the ammonia, and the key is how do we transport that ammonia safely in the bloodstream? Because free ammonia in the bloodstream is highly toxic. It can cause brain damage. If it travels, it can cross the blood-brain barrier and cause damage. It can cause a lot of problems. So we cannot have free ammonia traveling in the bloodstream. So the key to this slide, it's a little bit confusing, but what I want you to understand from this slide is the fact that that free ammonia is going to travel through the bloodstream in the form of glutamine. So we're going to take that ammonia that we just pulled off of glutamate, and we're going to transfer that ammonia to glutamine, or transfer that amino group to glutamine, and that is the way that we transfer ammonia. It's a non-toxic way to transport ammonia through the bloodstream. So glutamine is usually actually present in much higher concentrations in the bloodstream than other amino acids. We also know that it can serve as that amino group source in biosynthetic reactions. So when we're making our purines and permidines, we can use glutamine as that nitrogen source. So it can travel through the bloodstream. It can travel from place to place within the cell safely. And that is how we take that ammonia that is generated in extrahepatic tissue, so outside the liver, and that is the way that we allow that ammonia to travel to the liver so that it can be safely converted to urea and excreted. So that's the key to this chapter. Don't worry so much about everything else in this chapter, everything else on this slide. But what I really want you to know is that ammonia that we just extracted from glutamate on the previous slide, we are going to convert that or add that to glutamate to produce glutamine so that we can safely transport that ammonia through the bloodstream and ultimately to the liver to be converted to urea. So I'm going to stop there with this first half of chapter 18. So I'm going to upload this to Canvas, and then I will continue this lecture, recorded the second half of chapter 18 in another file.